Hi, and welcome back to my YouTube channel. By now you all know me, but if not, my name's Mario. I put up videos primarily on clinical research, and in today's video I'm going to talk all about ICH E9, the Statistical Principles for Clinical Trials. This video is part of my study series for the ACRP CCRA exam, or the Association of Clinical Research Professionals Certified Clinical Research Associate exam. So if you are studying for that exam or you want to learn about what CRA should know about statistics, definitely stay tuned. I'm going to cover that. I have the guidelines here. I'm going to just quickly go through them page by page and cover what I think is testable. The statistics guidelines are 43 pages long. So there's a lot of stats related stuff within this that I don't think is within the scope of a CRA's job or knowing them. So I won't spend too much time on things such as type 1 error, type 2 error, p-values, etc. If that's of interest to you, definitely read through the guidelines and learn more about the stats portion. What I'm really focused here on is what is testable for the CCRA exam. All right, before I dive into that, I need to remind you as always to take a moment and smash that like button and subscribe to my channel if you haven't done so already. Channel is almost at 2,000 subscribers, so thank you very much for your support. Much appreciated. All right, so before I go page by page, I just want to cover one concept, and that is something you should keep in mind whenever you're thinking about statistics. And that concept is reduce bias. You don't want to taint your results. So if you're struggling on a stats question, just keep in mind that whatever you're doing should probably reduce bias. You don't want to affect your results. So for example, it may ask you about the timing of when the statistical analysis plan should be written. And the answer is going to be before the trial starts. You don't want any data because if you had data, you could potentially introduce bias. So keep that in mind. That might guide you through the stats portion as an underlying principle. All right, so now let's get started. I'm going to flip through pages here and just quickly cover what I think is testable. Hopefully, I won't spend too long on this because 43 pages on stats is probably not really the idea of fun for most CRAs. So the first thing that comes here is scope and direction. In this section, it says the focus of this guidance is on statistical principles. Specific procedural steps to ensure that the principles are implemented properly are the responsibility of the sponsor. First testable question, who's responsible for the stats plan? It's the sponsor. So that is something as a CRA you should definitely know. The sponsor is also responsible for making sure that their statistician is appropriately educated and qualified. So those are the responsibilities of the sponsor. The next thing that I see is testable here is the timing, and I sort of alluded to this at the start. It says here, proposed statistical analysis should be clearly specified in a protocol written before the trial begins. So two things here, statistical plan should be built into the protocol, and it should be done before the trial begins. So primary relevant to clinical trials conducted in later phases. That's what this guideline is for primarily for later phase trials. And the substance of this document should be applied as far as possible to all phases of clinical development. The overall goal of these principles is to minimize bias and maximize precision. So those are the things that I think are the testable for the exam and general principle you should know. So talks about development plan in the next section, and I just think the only thing to know is that the broad aim of the process of clinical development of a new drug is to find out whether there's a dose range and schedule at which the drug can be shown to be simultaneously safe and effective to the extent that the risk-benefit relationship is acceptable. And then you want to make sure that this plan is written out clearly. So next section talks about two types of trials, confirmatory and exploratory trials. I think this is pretty important, so I'll spend a little bit of time here so you know the differences. So in general, I think the thing you'd really need to know as a CRA for the difference between the two is confirmatory trials can be used for efficacy, exploratory trials cannot. So if the trial is looking for an efficacy objective, then it's going to be a confirmatory trial that you're looking at. Exploratory is literally what the word says. It's to explore other things. So, as the guidelines put it, a confirmatory trial is an adequately controlled trial in which the hypotheses are stated in advance and evaluated. Confirmatory trials are necessary to provide evidence of efficacy or safety. In such trials, the key hypothesis of interest follows directly from the trial's primary objective. It's always defined, and the hypothesis that is subsequently tested when the trial is complete. So, confirmatory trials are intended to provide firm evidence in support of claims. Each trial should address only a limited number of questions. Just important in common sense. You don't want to ask so many questions that it becomes really hard to answer what you're asking. So you would see only one or two questions. So often it's safety, efficacy. Those are your primary objectives of a trial. So exploratory studies should have clear and precise objectives. In contrast to confirmatory trials, their objectives may not always lead to simple tests or predefined hypothesis. Analysis may entail detailed extrapolation. Such trials cannot be the basis of formal proof of efficacy. Note, exploratory cannot be proof of efficacy. That is important. So if you're looking for efficacy, confirmatory, and then exploratory explores other things. So I really think that's basically all that's testable here. And of course, it says the protocol should make clear distinction between the aspects of the trial which will be used for confirmatory proof and the aspects which will be used as exploratory analysis, which makes sense because you'd want to see in the protocol the difference in which aspects of a trial might be confirmatory, which might be exploratory. All right, the next section is scope of trials, talks all about populations. I don't think much is very testable here. So it says in your early phases of a trial, population may come from a very narrow subgroup, but as you get to the confirmatory trial stage, you want your subjects to mirror the target population. So, and I think the only thing I would note here is no individual clinical trial can be expected to be totally representative of future users. All right, the next section are primary and secondary variables. I think the only things to know here for the test are the definitions. So the primary variable should be the variable capable of providing the most clinically relevant and convincing evidence directly related to the primary objective of the trial. Generally be only one primary variable. So often safety, tolerability, quality of life, and health economics, those are primary variables. And then jumping here to the next page, it says it's critical to specify in the protocol the precise definition of the primary variable as will be used in the statistical analysis, clinical relevance of the specific primary variable selected, and the validity of the associated measurement procedures will generally need to be addressed and justified in the protocol. Primary variables should be specified in the protocol along with the rationale for its selection. The only thing I hear is the next sentence, which is redefinition of the primary variable after unblinding will almost always be unacceptable. This makes sense. Again, back to bias. You wouldn't want to be redefining primary variables after you see data. So that's just a concept to keep in mind. So secondary variables are either supportive measures related to primary objective or measurements or effects related to the secondary objectives. All right, the next sections I don't think are that testable, but I would just focus on the definitions. I'm not even going to go over them here. I'll just mention what they are. So composite variables, global assessment variables. I don't really, can't imagine that this is within the scope of a CRA job. So it goes on talking about multiple primary variables, surrogate variables, and categorized variables. And like I said, I would look at them, know the definition. I'm not covering them here because I couldn't come up with any potential questions that could be asked. So the next section is design techniques to avoid bias. The most important design technique for avoiding bias in the clinical trials are blinding and randomization. So how are the design elements that can, how do you design a protocol to reduce bias? You have elements of blinding and randomization. And these should be normal features of most controlled trials intended to be included in a marketing application. So then it breaks down the double blind approach, which is important. So no one involved in the conduct of the trial is aware of the specific treatment allocated to any particular subject, not even a code letter. So This is blinding. So there's, you should be familiar with blinding procedures, and there's single blind, there's double blind, there's actually triple blind. There's all sorts of things, but in most clinical research settings, you're going to be talking about a double blinded trial. You might also be talking about open label. And so I would spend some time on this section, and I'm going to read out blinding and randomization. So blinding or masking is intended to limit the occurrence of conscious and unconscious bias in the conduct and interpretation of clinical trials arising from the influence that the knowledge of treatment may have on the recruitment and allocation of subjects, their subsequent care, the attitudes of subjects to the treatments, the assessments of endpoints, the handling of withdrawals, the exclusion of data from analysis, and so on. So it says that a double-blind trial is one in which neither the subject nor the investigator or sponsor staff involved in the treatment or clinical evaluation of the subject are aware of the treatment received. So that is your definition for a double-blind study. In a single-blind trial, the investigator and his or her staff are aware of the treatment, but the subject is not, or vice versa. In an open label trial, the identity of treatment is known to all. So in a single blind trial, the investigator and or his staff are aware of the treatment, but the subject is not, or vice versa. So one side is aware, one side is not. And then open label trial, treatment is known to all. So it talks about here difficulties in achieving the double blind ideal can arise. It talks about something called double dummy, which it says this technique may sometimes force an administration scheme that is sufficiently unusual to influence adversely the motivations and compliance of the subjects. I don't think you need to know about double dummy. So if a double-blind trial is not feasible, then the single-blind option should be considered. In some cases, only an open-label trial is practically or ethically possible. All right. Now, I have, I've starred this section because I think this is really important as a CRA, and this is breaking the blind. So this is something you should be very aware of. So I'm going to take some time, just read through it here. Breaking the blind for a single subject should be considered only when knowledge of the treatment assignment is deemed essential by subject's physician for the subject's care. Any intentional or unintentional breaking of the blind should be reported and explained at the end of the trial, irrespective of reason of its occurrence. So when you're designing blinded trials, you want to make sure there's an easy way to break the blind, because if the subject ends up in an emergency situation, you want to make sure you can pretty much find out the treatment assignment quickly. However, you don't want to do this in all cases. It's only if it's needed for the subject's care. So that's specified here. Just keep that in mind. You don't always have to break the blind, but you want an easy way to break the blind if it's needed for someone's care urgently. So next section is randomization. Randomization introduces a deliberate element of chance into the assignment of treatments to subjects in a clinical trial. In combination with blinding, randomization helps to avoid possible bias in the selection and allocation of subjects arising from the predictability of treatment assignments. Randomization schedule of a clinical trial documents the random allocation of treatments to subjects. And then it says, although unrestricted randomization is acceptable approach, some advantages can generally be gained by randomizing subjects in blocks. All right, so that's randomization for you. It says here in multi-center trials, the randomization procedure should be organized centrally, and the next subject to be randomized in trial should always receive the treatment corresponding to the next free number in the appropriate randomization schedule. The randomization schedule itself should be filed securely by the sponsor or an independent party in manner that ensures the blindness is properly maintained throughout the trial. So who does the randomization schedule and maintains it? It should be done by either the sponsor or an independent party, and make sure that the blind is appropriately maintained. It says dynamic allocation is an alternative procedure in which the allocation of treatments to a subject is influenced by the current balance of allocated treatments, and in a stratified trial by the stratum to which the subject belongs and the balance within the stratum. All right, moving on to the next section, which is trial design considerations. So I feel like this is something that I didn't necessarily know the terms, but I knew as a CRA. So I would spend time knowing your design configurations of protocols. So specifically the parallel group design, crossover design, and factorial design, which are the three types of potential trial configurations. So I'll just go over the definitions and we'll move on, since this is something that you're going to be familiar with if you read protocols, but you might not necessarily have known the term. So it says the most common clinical trial design for confirmatory trials is the parallel group design, in which subjects are randomized to one or two or more arms, and each arm being allocated for a different treatment. So this is your protocol schema you might have seen with different randomization arms. So that is a parallel group design. So crossover design. In the crossover design, each subject is randomized to a sequence of two or more treatments and hence acts as his own control and treatment comparison. This simple maneuver is attractive primarily because it reduces the number of subjects and usually the number of assessments needed to achieve specific power, sometimes to a marked extent. If you think about it, though, if you're using less subjects and there's less subject compliance, that's a bigger problem, but that is what your crossover design is. So it says when the crossover design is used, it's important to avoid carryover. Crossover design should generally be restricted to situations where losses of subjects from the trial are expected to be small. All right, so I just mentioned that is a concern when you're using less subjects. All right, factorial design. In factorial design, two or more treatments are evaluated simultaneously through the use of varying combinations of treatment. So those are your things. So like I said, just know what they are. I think that's the only way I could see this tested, is it gives you some scenario and asks is what kind of a design. Is this parallel design? Is this factorial design? Or is this crossover design? All right, so next section is on multi-center trials. So multi-center trials are carried out for two main reasons. Multi-center trial is an accepted way of evaluating a new medication more efficiently, and a trial may be designed as a multi-center trial primarily to provide a better basis for the subsequent generations of its finding. So that's just what you need to know. That's why we do multi-center trials. Says here, good design should generally aim to achieve the same distribution of subjects to treatments within each center, and good management should maintain the design objective. In most instances, certain centers can be satisfactorily defined through the investigators. All right, so that is all just why we do multi-center trials. I think that's basically all you need to know. And then it has the type of comparisons. This is jumping a little bit into the stats, but I think things that are probably good to know. So the types of comparisons you can do are trials to show superiority, trials to show equivalence or non-inferiority, and trials to show dose-response relationships. And that is it. So those are the types of comparisons. Again, I think definitions here and move on. Trials to show superiority. So scientifically, efficacy is the most convincingly established by demonstrating superiority to placebo in a placebo-controlled trial, by showing superiority to an active control treatment, or by demonstrating a dose response. So the appropriateness of placebo control versus active control should be considered on a trial-by-trial basis. And it says there, for the equivalence or non-inferiority, in some cases, an investigation product is compared to a reference treatment without the objective of showing superiority. This type of trial is divided into two major categories: equivalence and non-inferiority. So it'll break down the different things for equivalence and non-inferiority. So many active control trials are designed to show that the efficacy of an investigation product is no worse than that of an active comparator. So that's showing non-inferiority. Bioequivalence trials fall into the former category of equivalence. So you're looking at bioequivalence trials. So I can't imagine much being tested here. So just cover active control equivalence and non-inferior trials may incorporate a placebo, thus pursuing multiple goals in one trial. Active comparators should be chosen with care. It's vital that the protocol of a trial designed to demonstrate equivalence or non-inferiority contain a clear statement that this is explicit intention. So again, things that you should write in protocol. Just covering this, statistical analysis is generally used based on the use of confidence intervals. For equivalence trials, two-sided confidence intervals should be used. This is where I was talking about. This talks about type 1 errors as such. This is well beyond the scope of what I'd expect a CRA to know. So trials to show dose-response relationship. How response is related to the dose of a new medicinal product is a question to which answers may be obtained in all phases of development and by a variety of approaches. Dose-response trials may serve a number of objectives: the confirmation of efficacy, the investigation of the shape and location of the dose-response curve, the estimation of an appropriate starting dose, the identification of optimal strategies for individual dose adjustment, the determination of a maximal dose beyond which additional benefit would be unlikely to occur. So dose-response relationships, that's what you need to know. And I think that's basically all for that section. Talks about group sequential designs. I actually didn't see anything testable here. Sample size. The number of subjects in a clinical trial should always be large enough to provide a reliable answer to the questions addressed. The number is usually determined by the primary objective of the trial, and then it goes well into that. So I think that's really all you need to know. You want to make sure you have enough subjects to meet your primary objective. So sample size calculation should refer to the number of subjects required for the primary analysis. Moving on to the next section, data capture and processing. The collection of data and transfer of data from the investigator to the sponsor can take through through a variety of media, including paper, case report forms, remote site monitoring systems, medical computer systems, and electronic transfer. Whatever data capture instrument is used, the form and content of the information should be in full accordance with the protocol and should be established in advance of the conduct of the clinical trial. So I think you're seeing the recurring theme. Things need to be decided in advance. Trial considerations, it talks about trial monitoring and interim analysis. This will likely be covered when I talk about ICH E6 later on. So I'll just cover a sentence or two here. And it says, careful conduct of a clinical trial according to the protocol has a major impact on the credibility of results. Careful monitoring should ensure that difficulties are noticed early and their occurrence or recurrence minimized. Sorry. Let me read that again because I read that quickly. So careful monitoring can ensure that difficulties are noticed early and their occurrence or recurrence minimized. So this is the things like deviations, which we notice and try to minimize them as they go on. So it talks about two distinct types of monitoring. One type of monitoring concerns the oversight of the quality of the trial, while the other type involves breaking the blind to make treatment comparisons. So for the purpose of overseeing the quality of the trial, the checks involved in trial monitoring may include whether the protocol is being followed, the acceptability of data occurred, the success of planned accrual, the appropriateness of its design assumptions, the success in keeping patients in the trials, and so on. The period for this type of monitoring usually starts with the selection of trial sites and ends with the collection and cleaning of the lost subjects' data. So it talks about monitoring. Like I said, this is going to be something that's going to be covered in a video that I should put out and plan to put out in the future about ICH E6, which would cover a lot of this. Talks about changes to exclusion and inclusion criteria. Inclusion and exclusion criteria should remain constant as specified in the protocol throughout the period of subject recruitment. Changes should be made without breaking the blind and should always be described by a protocol amendment. So you want your inclusion and exclusion criteria to be set out up front. There might be changes needed, and if you do, and oftentimes this is because you put in something very restrictive, such as some inclusion criteria that really impacts your ability to recruit. So the way to do that, then, if you change your inclusion exclusion criteria to impact your recruitment, is to do so with the protocol amendment. It talks about accrual rates in trials with a long time scale for the accrual of subjects. The rate of accrual should be monitored. Talks about sample size adjustments. The potential need for re-estimation of the sample size should be envisaged in the protocol whenever possible. Interim analysis and stopping. rule. So an interim analysis is any analysis intended to compare treatment arms with respect to efficacy or safety at any time prior to the formal completion of the trial. All interim analysis should be carefully planned in advance and described in the protocol. Special circumstances may dictate the need for an interim analysis that was not defined at the start of the trial. In these cases, a protocol amendment describing the interim analysis should be completed prior to unblinded access to treatment comparison data. So protocols should describe the schedule of interim analyses. The stopping guidelines and their properties should be clearly described in the protocol or amendment. The execution of an interim analysis should be a completely confidential process because unblinded data and results are potentially involved. Investigators should be informed only about the decisions to continue or discontinue a trial or to implement modifications to trial procedures. So trial should be stopped early only for ethical reasons or if the power is no longer acceptable. And this highlighted here for me, for many clinical trials of investigative products, especially those that have major public health significance, the responsibility for monitoring comparisons of efficacy and or safety outcomes should be assigned to an external independent group, often called an Independent Data Monitoring Committee, or IDMC, a Data and Safety Monitoring Board, or a Data Monitoring Committee. So the next section talks about the roles of the IDMC, or Independent Data Monitoring Committee. It says an IDMC may be established by the sponsor to assess the intervals, at intervals, the progress of a clinical trial, safety, data, and critical efficacy variables, and recommend to the sponsor whether to continue, modify, or terminate the trial. Just note that it says IDMC here, and it clearly says their role, and I'll read this again, is for to assess progress of a clinical trial, safety data, and critical efficacy variables recommended by the sponsor, and whether to continue, modify, or terminate the trial. The one thing that an IDMC doesn't do is whether or not the trial should be initiated. That's not within the scope, and I can see that being a testable question. They will throw in, is the IDMC responsible for starting the trial? And the answer is no. It looks at the safety and the continuation and possibly termination, but it does not give a green light on whether or not to start the trial. That's not the role of the IDMC. So whether a sponsor representatives on the IDMC, their role should be clearly defined in the operating procedures of the committee. I think that's that section basically, and then it talks about pre-specifications of the analysis. So the statistical analysis plan may be written as a separate document to be completed after finalizing the protocol, but the plan should be reviewed and possibly updated as the result of the blind review of the data and should be finalized before breaking of the blind. And just started off with when designing a clinical trial, the principal features of the eventual statistical analysis of the data should be described in the statistical section of the protocol. Talks about analysis sets, the set of subjects whose data are to be included in the main analysis should be defined in the statistical section of the protocol. Content, but it should, at minimum, should be demographic and baseline data on disease status should be collected whenever possible. So frequency and type of violations, missing values, and other problems should be documented in the clinical study report, and their potential influence on the trial should be described. So if there are violations or things missing, you would expect them to be described in the clinical study report or the CSR. That could potentially be a testable question. All right, so looking now into the next portion, which talks about full analysis set. And this is something to really know the definitions for. So it talks about the intention to treat principle implies that primary analysis should include all randomized subjects. So this says intention to treat. So it doesn't even mean that the subject might have necessarily been treated, but they were intended to treat. So the intention to treat group, it includes all randomized subjects. So and it talks about a full analysis set is used to describe the analysis in which is as complete as possible and close as possible to the intention to treat ideal of including all randomized subjects. And then it talks about there are a limited number of circumstances that might lead to excluding randomized subjects from the full set analysis, including failure to satisfy major entry criteria, the failure to take at least one dose of medication, the lack of any data post-randomization. So I think that's the thing you need to know. The intention to treat includes subjects that you intended to treat, and later, your full set will include all those subjects, and then later you can eliminate things like subjects that didn't receive drug, etc. So when the full analysis set of subjects is used, violations of the protocol that occur after randomization may have an impact on the data and conclusion, particularly if their occurrence is related to treatment assignments. Don't see here. Okay. The next section here is the per protocol set. And again, definition. This is a recurring theme for stats. You should know these definitions, but I wouldn't dig too much beyond knowing the definitions. What is a per protocol set? The per protocol set of subjects, sometimes described as valid cases, the efficacy sample, or the evaluable subject sample, defines a subset of the subjects in the full analysis set who are more compliant with the protocol, and is characterized by criteria such as the following: the completion of certain pre-specified minimal exposure to treatment regimen, the availability of measurements of the primary variable, the absence of any major protocol violations, including violation of entry criteria. So the precise reasons for excluding subjects from the per protocol set should be fully defined and documented before breaking the blind in a manner appropriate to the circumstances of the specific trial. So if you're going with the per protocol set, so this subset, you should define it before you break a blind. So that makes sense. It talks about the roles of different analysis sets. It's advantageous to demonstrate lack of sensitivity of the principal trial results to alternative choices of set of subjects analyzed. So, all right. So it talks here a little bit about missing values and outliers. Missing values represent a potential source of bias in a clinical trial. Hence, every effort should be undertaken to fulfill all the requirements of the protocol concerning the collection and management of data. There will always be some missing data. So this regulations acknowledge there will always be some missing data. Talks about data transformation. In the next, the decision to transform key variables prior to the analysis is best made during the design of the trial on the basis of similar data from earlier clinical trials. Talks about estimation, confidence intervals, and hypothesis testing. So this is jumping up into the more stats portion. So the statistical section of the protocol should specify the hypotheses that are to be tested and or treatment effects that are to be estimated in order to satisfy the primary objectives of the trial. The statistical methods to be used to accomplish these tasks should be described for the primary and preferably the secondary variables, and the underlying statistical model should be clear. So it says within the statistical section of the protocol or the statistical plan, there should also be an outline of the way in which data other than the primary and secondary variables will be summarized and reported. So then it talks about adjustment of significance and confidence intervals, subgroups, interactions, and covariance. So this is jumping more into things that I don't think are really testable for the ACRP CCRA exam. So I'll just go over what I've underlined here. So the primary variable is often systematically related to other influences apart from treatment. In some instances, adjustment to the influence of covariates or for subgroup effects is an integral part of the planned analysis and hence should be set out in the protocol. And it's not advisable to adjust the main analysis for covariates measured after randomization because they may be affected by treatments. All right, the next section here is the integrity of data and computer software validity. You just want to make sure that you have validity checks of your software. That's what the whole section basically states. Then goes into evaluation of safety and tolerability. So in all clinical trials, evaluation of safety and tolerability constitutes an important element. Talks about the choice of variables. In any clinical trial, the methods and measurements should be chosen to evaluate the safety and tolerability of the drug will depend on a number of factors, including knowledge of the adverse effects of closely related drugs, information from non-clinical or earlier clinical trials, and possible consequences of the PD, PK properties of the particular drug, the mode of administration, the type of subjects to be studied, and the duration of the trial. And then it says here it's recommended that a consistent methodology be used for the data collection and evaluation throughout a clinical trial program to facilitate the combining of data from different trials. The use of a common adverse event dictionary is particularly important. And I think that would be covered in a different section of regulations, but just know that the adverse events, generally you want to put them under dictionary terms, and we have a common adverse event dictionary that exists. So we have things such as the preferred term for an adverse event. All right, so it says set of subjects to be evaluated and presentation of data. So for the overall safety and tolerability assessment, the set of subjects to be summarized is usually defined as those subjects who received at least one dose of the investigational drug, and we're looking at things such as type of adverse event, severity, onset, duration. All safety and tolerability variables will need attention during evaluation, and broad approach should be indicated in the protocol. All adverse events should be reported whether or not they're considered related to treatment. And then, it goes on here to the statistical evaluation. The investigation of safety and tolerability is a multidimensional problem. The range of possible adverse effects is very large, and the new and unforeseeable effects are always possible. An adverse event experienced after a protocol violation, such an excluded medication, may introduce bias. Again, this is where if you're doing things that aren't in the protocol, you can potentially introduce bias. It talks about the integrated summary. The overall usefulness of a drug is always a question of balance between risk and benefit. And then it says the reporting for the evaluation and reporting section. During the planning phase of a trial, the principal feature of the analysis should have been specified in the protocol. And it talks about blind decisions will generally introduce less potential bias. So statisticians or other staff involved in unblinded interim analysis should not participate in the blind review or in making modifications to the statistical analysis plan. This is a potentially testable concept. Just know that for blind decisions, you don't want people that are unblinded to be involved in making modifications to the statistical analysis plan. Again, you're introducing bias. All right, all subjects who are entered in a trial should be accounted for in your report, whether or not they are included in the analysis. All reasons for exclusion should be documented. Subjects lost to follow-up, withdrawn from treatment, or with a severe protocol violation should be identified, and a descriptive analysis of them provided, including reasons for their loss and its relationship to treatment outcome. And it says, although the primary goal of the analysis of a clinical trial should be to answer the questions posed by its main objective, new questions based on the observed data may well emerge from the unblinded analysis. And now we're getting to the last part here. And it says, sparing use should be made of unplanned analysis. So the trial statistician should be a member of the team responsible for the clinical study report and should approve the clinical report. Again, testable question. How is the statistician involved in the final report? And they actually need to look at and approve it. So that is part of the statistician's job. This is going back all the way back to the beginning, that the sponsor is responsible for picking a qualified statistician. So then it says summarizing the clinical database. An overall summary and synthesis of the evidence on safety and efficacy from all the reported clinical trials is required for the marketing application. This may be accompanied when appropriate by a statistical combination of results. Specific statistical interests arise, describing the demography and clinical features of the population treated during the course of the clinical trial program, addressing the key questions of efficacy by considering the results of the relevance, usually controlled trials, and highlight the degree to which to reinforce or contradict each other, summarizing the safety information available from a combined database of all the trials, whose results contribute to the marketing application, and identifying potential safety issues. So this is just summarizing your clinical database. Next section is just efficacy data. Individual clinical trials should always be large enough to satisfy their objectives. And then safety data. In summarizing safety data, it is important to examine the safety database thoroughly for any indications of potential toxicity and to follow up on any indication by looking for associated supportive patterns of observation. All indications of potential toxicity arising from the exploration of the data should be reported. The risks associated with identified adverse events should be appropriately quantified to allow proper assessment of the risk-benefit relationship. And that brings us to the last section, which is the glossary of terms. I'm not going to go over this. Everything in theory was covered earlier. So that is everything you need to know about stats. If you stayed with me this long, I appreciate it. This is a really long video. I know stats is actually fairly boring and not a huge thing I think about on a day-to-day basis, but is really important. All right, if you made it this far, please take a moment and smash that like button, and I will see you in the next video in which I will cover ICHE 8. Thank you for watching.